Like no pockets or anything in this, except for that. $40. So that's not great for the actual test. Sucks. And that came out and I was like, what is that? Yeah, those ones trip me up. I really think that's where I'm like 2 weeks of this yeah. So. Yeah, I should have done that before I got quiet. Yeah, I don't still have the notes, so after this I'll have to go to the library. Yeah. In all seriousness, as long as I know how to join a level, I won't do that like nursing club here at severe contract, like not at or is it like more? We have one son.
Okay, let's go ahead and start. Zoom surprisingly decided it wasn't going to work, and all of a sudden it decided it was. So I can't take any credit for it, but it somehow is working. So I'm just going to say thank you and go on from there. So we are just about done, if I remember correctly, since there wasn't a video, because if Zoom doesn't work, I don't get a video to record. I'll just tell you that. We were just about done with the chemistry chapter. I think all we had to do was look at, I think we'd finished the DNA and RNA, if I'm right. Everybody nodding their head. Nod your head or shake your head, just make me feel better. So anyway, that'll do. Don't get dizzy. I think we're just about to talk about the ATP, and then we can go on into chapter 3 where we start talking about cells. So if I'm wrong, somebody tell me, but I think that we'd finished up the DNA and the RNA. So, okay, good. So now let's go into chemistry and the last 2 slides. That should be just what we want. So, okay. Okay, technology is wonderful when it works. Just a reminder that when we start lab this afternoon, we're going to be looking at the appendicular skeleton and joints and articulations. So we're going to go on through that. The appendicular skeleton is the arms and legs, hands and feet and stuff like that. So the beauty of that is whatever you have in one side of your body, you have on the other side. So that's what the lab is for AP1 this week. Going to be the appendicular skeleton. So as soon as I finish this section up here, we're going to go into chapter 3 and start looking at the cells and the cell structure. So the very last thing under the nucleic acids in chemistry is what we call ATP. Now it's not really a nucleic acid like DNA and RNA is. ATP is what our cells use to transfer energy around. We get energy from glucose. Everything we eat is converted to glucose. And then we will oxidize. We'll actually, it's sort of like we're burning glucose. We're oxidizing it. We're releasing the energy that's in the covalent bonds in the glucose. We can't release all that energy at one time. There's a lot of energy in a molecule of glucose. We don't want to release that. So what we do is we release it gradually and we dump the energy off into these molecules called ATP. And it's ATP that we send around the body to supply our energy needs. We make ATP usually where we need it. We make a lot of ATP in our legs and our arms, and our skeletal muscles need energy. So we were producing a lot of ATP in our legs and arms. Skeletal muscle has a lot of glycogen stored there. That's how they get their sugars. Glycogen is converted to glucose, and then the glucose is oxidized. It's burned, if you will. And that energy, instead of releasing it all at once, which would really be bad for us if all that energy came out at once, think the Hindenburg. Everybody remember the Hindenburg from history? Imagine us, the concept of spontaneous human combustion could actually occur if all that energy in a molecule of glucose was released at one time. So we don't do it that way. We release it into ATP. And ATP is how we power everything, the contractions of our muscles, the nerve impulses that go through our brain and spinal cord, the contraction of our heart, the squeezing of food through our digestive tract, the movement of substances across the membrane against the concentration gradient. Things that we really want to get into the cell, but it's not going to go by diffusion. We spend energy in all that stuff and we release it from the glucose constantly. The problem with ATP is that it only lasts for 7 seconds. So we can't store it. We can make it and use it, but we can't store it. So we have to constantly make new ATP. It's a very simple molecule. The reason we throw it in here with the nucleic acids is it's made of a nucleotide. Remember what a nucleotide is? A nucleotide has a base, a sugar, and a phosphate. DNA is made of nucleotides. RNA is made of nucleotides. DNA and RNA use the nucleotides, the sugar and the phosphate as their backbone, like the rungs of a ladder. Not the rungs, but the sides of a ladder are the sugars and the phosphates, and the bases are the rungs. Well, ATP is a lot simpler. It's just the nucleotide. It's a nucleotide that consists of a 5-carbon sugar, a base called adenine, and 3 phosphate groups. It looks something like this down there at the bottom. Adenine is attached to the ribose sugar. And on the other side of the ribose sugar is where the phosphate's attached. And here, we instead of having 1 phosphate like we see in DNA and RNA, we have 3 phosphates. The 3rd phosphate on here is unstable. We have to force it on. We have to use energy to force it on. Who hasn't driven a nail or forced a screw into something and who hasn't used a hammer at some point to get a screw to go in because it won't go in right, when you're in a hurry. Not supposed to, but we've all done something like that, there are all sorts of bizarre things that we've all done. However, the third phosphate has to get forced. It takes a lot of energy. And when you force that third phosphate onto the molecule, that energy stays in there for 7 seconds. And so instead of sending glucose around, we can send ATP around to where we need it. And it'll last for 7 seconds, and then it'll break down. It'll break apart to what we call adenosine diphosphate, which is the 2-phosphate version, and what we call inorganic phosphate. And as soon as it breaks apart, then if we have energy available, we're going to force it back together again. We don't really ever destroy ATP. We just keep making it over and over and over again. And this is the currency, the money, if you will, our cells use to power things up. We're always making ATP. We use glucose as the source of our energy, and we convert it to ATP so we can send it around. So we can control how much we're making, because if we were to release all the energy in a molecule of glucose, it would be outstanding. Very briefly outstanding. There's a lot of energy in a molecule of glucose, and we want to release it in a controlled way. So, okay. That is the end of chapter 2, which means we've finally finished unit 1. Now, the pace is going to pick up a little bit as we go further on. We won't have as much to cover in each of the following 2 chapters, but we're going to have a lecture test sometime soon. That's just the way it is. So let's see. I was thinking to give you plenty of time to get ready for your first lecture test. I would think that it would open up next Thursday. It'll give you a little over a week. A week and to give you 9 days to get ready for your lecture test. So we don't have anything coming up in lab immediately, so you don't have to worry about that. But we can get the lecture test out of the way. And yes.
Do you have any study guides like you did for the lab?
Yes, they are posted in the content of eLearn. There are some very detailed study guides for chapters 1 and 2 in there. Yes. You have the option. You can always come in here and take it. You can take it down with the tutor, or you can take it online. So every test will be different. They're all randomized. So, but yeah, you have the option of taking it 3 different ways. Or actually 2 different ways. I would, it doesn't matter to me. Whatever is more convenient for you all. If you're going to take it online, I'll open it up next Thursday and leave it open until Friday night right before midnight. You'll have a 48-hour window to take the test. And if you had issues with the lab test, that I'm pretty amenable to extending times if you need it. If you have a good, if something happens, so don't worry about it. Just send me an email and I can fix it. But we'll plan on that then next Thursday. Plenty of time. Now, what we're starting today on chapter 3 on cells, and the next chapter, which is 24, I know it's way out of sequence, but that's how we do it. The chapter on metabolism, I'll leave it like that. That's unit 2. That'll be its own separate test. So nothing on here from here on today is on the test coming up next week. Okay, let me get out of this. Let me go in, pull up the PowerPoints. Okay, hopefully this is the right set. Here we go. And you should all see something that looks like what's on the screen here. And if you're zooming, you should all see it says chapter 3, cellular level of organization. So, okay, first slide. Cells. Cells are what make us up. We have billions of cells in our bodies. Every living organism is made of cells. You can't be alive and not have cells, even if you only have one cell. You know, some organisms are only one cell. We've got lots of one-celled organisms, you know, parameciums and different types of bacteria. You know, an amoeba is a one-celled organism. But everything that's alive is made up of at least one cell or billions of cells like us. Cells, the second thing about part of the cell theory, cells are the basic living unit of structure and function in organisms, meaning that, sort of like I said, living organisms are made of living cells. There's nothing smaller than a cell that's alive. So a cell is the smallest living unit we encounter. And the third point here is that living cells only come from other living cells. No matter what you may have read recently about some work done in Israel with artificial cells, those are not true living cells. I don't know a whole lot about what they did, but they're claiming that they created artificial cells that are alive. Well, they may be acting, they may be reacting, but they're not living cells. Living cells can only come from other living cells. And they can survive in a variety of mediums, not necessarily in a laboratory. You know, there's no such thing as proto-life on the shelf that we can open it up and pour it out into a beaker and, oh, we've got life. It doesn't work that way. And you know what's amazing is that this is not a testable point, but what's amazing, it took up until the middle of the 1800s, the 19th century, you know, in the 1850s, before scientists had fully developed the idea that all plants are made of cells and all animals and humans are made of cells. So that idea has only been around for about 150 years, you know. So, you know, early scientists believed that life spontaneously arose, you know. So, okay. Now, how many types of cells we have in our bodies? We have over 200 different types, and we have different sizes and shapes and structure and functions. The structure is important for the function. You know, we have long, skinny nerve cells that stretch out for 3 or 4 feet in our bodies because they have to go from the spinal column all the way down to our big toe. That cell is designed to stretch that far. Red cells are designed to be what we call a biconcave disc, meaning it's like a big water balloon, a squishy water balloon that has a dimple in the top and the bottom side of it. It looks sort of like a donut that you sort of squeeze the middle on. Red cells are designed that way so they can have more surface area to carry out to collect oxygen. The muscle cells are designed to contract, to slide over. The components of the cell are designed to slide over each other. Every cell is different. But one thing they all have in common is that they either have a nucleus with the DNA or they had a nucleus with their DNA, because the red cells are the only ones that kick their nucleus out. So, okay. So if you're a cell, and you probably heard me say this in lab, every living cell in our bodies, except for red blood cells, have a plasma membrane. That's the outer covering. You all saw where your plasma membrane is located when you did the cheek cells in lab. Every cell has cytoplasm. That's the goop inside the cell membrane. And every cell has a nucleus, except again, red blood cells kick theirs out late in their formation. Red cells would rather only last for 4 months than have a nucleus that takes up space where they can carry oxygen. So, but all the other cells in our bodies are going to have these three parts: a plasma membrane, which is the outer boundary, the cytoplasm, where all the organelles, the little smaller structures within the cell float. It has a consistency, and we've called it cell sap. We've called it, protoplasm. We've called it cytoplasm. We've called it cytosol. It's the material that holds the organelles. Okay. Now, I hope that's not my doctor's office. No, it wouldn't. Okay. Now, cell membrane is a barrier. The cell membrane is what we call a selectively permeable membrane. It is made up of phospholipids, so it has this arrangement that we saw when we talked about lipids, where you have a phospholipid has a phosphate head and a couple of tails. We have the fatty acid tails, and we have the phosphate head. And the cell membrane is made up of a double layer of these structures. So what's known as a phospholipid bilayer. The cell membrane keeps the contents of the cell separate from the outside. The interstitial fluid that surrounds every cell, that comes, interstitial fluid is the fluid that is surrounding our cells, that came from plasma. Blood vessels leak all the time. Plasma, the fluid medium that blood floats in, the red cells and white cells float in, is always leaking out. We call it interstitial fluid, meaning interstitial means around the cells, on the outside of the cells. And so then we call that the extracellular fluid or interstitial fluid. And we keep that out of the cells. The cell membrane protects the fluid inside the cell from the fluid outside. So it also restricts the movement of substances across the membrane. It doesn't let water across. It doesn't let proteins across the membrane. It doesn't let anything across the membrane except something that is soluble in fat. There are some exceptions. We'll talk about those. So that's the plasma membrane. It does a very good job of keeping the cell separate from every other cell and the contents of what's outside. So it's very flexible. It's like, imagine if you had a squishy, it's very flexible. Imagine if you had a squishy water balloon covered in olive oil. So that's your olive oil makes it waterproof, and yet it can move up and down and around. And, water balloons, they change the shape of your hand, how you're holding them. You're gonna, and they change when they hit. They go from round to they break apart and they splatter. But a water balloon is a very squishy structure, and that's a lot like our cells. Our cells can change their shape and be very flexible as they need to, and that's because of the membrane. So the membrane has lots of structures embedded there. We end up calling the membrane a fluid mosaic because of all the proteins and the carbohydrates and the cholesterols embedded in there. We have a structure on the surface of the cell called the glycocalyx. The glycocalyx is our identifier. Every cell in our body has a glycocalyx. It's made of a protein and a carbohydrate, and it sticks up and says, hey, it's me. And it's telling your immune system not to kill your cell. So the glycocalyx structures in all of our cells are identical, identifying us to our immune system as belonging to us. We also have places where the cells can join together. We have junctions that hold the cell together or have openings so that cells can communicate. So, let's see.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions, such as sodium ions and hydrogen ions, cannot pass through the membrane unaided.
Okay, the term to describe our cell membrane is a phospholipid. I hope I spelled that right. Thank you. Bilayer. Phospholipid bilayer, because it's a double layer of these phospholipids. And as you can see, unless you're fat soluble, nothing's getting in. Now, gases like CO2 and oxygen are so small, they can, the molecules are so small, they can diffuse across the layer. Water can't get in without help. Sodium and potassium can't get in or get out without help. Glucose needs help to get in into the cell. So while it is things, it is selectively permeable, meaning it lets some things in, like things dissolved in fats, like hormones that are made from cholesterol, and it restricts other things, but there are still ways to get in and out of the cell. Cells still need water. Cells still need glucose. Cells still need sodium and potassium. So there are specialized mechanisms to get across the membrane. But most everything else has a real hard time getting in. Most things won't get in. So it's part of the way the cell protects itself. Okay. So there's your phospholipid. Well, it looks a lot nicer than the one I drew. But that's a phospholipid. Your phosphate, it's sort of like a triglyceride. It still has glycerol, the 3-carbon glycerol, but instead of having 3 fatty acid chains, it only has 2. Where we would have the 3rd fatty acid, we would have a phosphate molecule. And so we get a different shaped structure. We don't get the we don't get the triglyceride. We get this, where we have the phosphate head and the fatty acid tails. And the fatty acid tails are not going to stick together like solid fats do, because the fats are going to be unsaturated. So van der Waals will keep them from kinking, solidifying. But they, the fatty acid tails, what happens when the oil and water mix? Well, rather, what happens when oil and water don't mix, actually. We can't mix oil and water, right? If you have like Italian salad dressing, what do you have to do before you use it? You have to shake it up, because the oil is in the top and the vinegar and everything else is underneath with all the seasonings. You have to shake it up. But even then, you're not mixing the oil and water together. You're just going to break the oil apart long enough to get the oil and the vinegar or whatever you're having on your salad. Well, it's the same thing with our cell membrane. Water doesn't get across. Anything suspended in water doesn't get across. Large molecules don't get across. Hormones, chemical messengers, most of them are made of proteins. Not one of those can get across the membrane. The only hormones that can get across the membranes are the ones based off of cholesterol. So anyway, and there's what it looks like again. So you have the hydrophobic center, meaning the water-hating center, and you have the phosphates on the outside. So now the cell membrane looks a lot like this. It's a very busy location. Imagine it's moving up and down. It's like you go into a bounce house. If you've ever in a bounce house when you were a kid, or if you got kids that have gone in a bounce house and they actually found their way out, the surface is soft and springy and it always moves up and down. That's what our cell membranes are like. They're constantly in motion, up and down. There are proteins that are sticking all the way through the membrane. There are rafts of cholesterol floating on the membrane. There are proteins and carbohydrates stuck together, sticking out. We have carbohydrates and fats, sort of a funnel cake of the cell, because it's the glycolipid up there in green is a sugar-coated fat. So I call it the funnel cake of the cell, with apologies to funnel cake. Anyway, we have glycoproteins, which are our identifiers, and we have glycolipids, and then we just have cholesterol sticking through, we have carbohydrates sticking through, and the whole thing is moving up and down the whole time. So it's sort of like, back to my water balloon, it's a water balloon coated in oil, rolled in cereal, and it's constantly in motion. The cereal is sliding across the surface, like, let's say it's Cheerios. The Cheerios are sliding across the surface of the cell the whole time. It's constantly moving up and down, and everything's floating on the oily surface and sliding. And that's sort of what a cell is like. A cell is a lot more sophisticated in its design, but that's how it works. It's not this rigid, firm structure that never changes. It's always in motion. The cell uses its membrane as a workshop or a laboratory or a passageway for things to get in or things to get out, because the cells are always making things too. Cells are always making proteins, and they're releasing those proteins from the cell membrane. They're making antigens. They're making antibodies to attack antigens. They are making lysosomes to fight infection. Those are organelles that are going to release that contain digestive acids to break down things. So cells are very, very busy, and they had to release stuff. They also have to get stuff. So while this membrane is selectively permeable and it's real squishy, it does let stuff in and out. Yeah. Okay, if you're interested in numbers, it's 3/4 phospholipids, 5% glycolipids, the sugar-coated fats, and 20% cholesterol. So someone who tells you that having zero cholesterol in their diet is a great idea is wrong. We need cholesterol. We need a certain amount of cholesterol and a certain amount of fat in our diet. We don't have to have a lot, but we do have to have some cholesterol and some fats because we need to use them. We need to make hormones. We need to keep the cell membrane intact. We need to make cholesterol. Now, the problem we get into, of course, is that we have too much cholesterol in our diet. So within the cell membrane, arguably the most important structures in the cell membrane are the proteins. The proteins allow us to communicate. Proteins allow us to communicate from the outside in and the inside out. Proteins allow us to connect cell to cell so they can communicate rapidly. Some proteins can go all the way through the membrane. Some are just on the surface, the top surface, bless you, or the inside surface. Constantly in motion. The two types of proteins we have are what are known as the integral proteins that go all the way through, or the peripheral proteins, which usually hug the inside of the membrane. They can be on the surface, but many of them hug the interior surface too. So integral proteins, they are what we call transmembrane, which is a nice way of saying they go all the way across. These proteins function as carrier proteins or channel proteins. They're transport. A channel protein is a tube, forms a tube from the outside to the inside. That's how we can get water into a cell. Channel protein forms and water comes through and the cell can get the water it needs. It's been suggested that, actually it's probably more than suggested, but the role of insulin, we all know, if you eat something with carbohydrates and your blood sugar goes up, your body releases insulin so the sugar can go into the cells, right? We've mentioned that a couple times. How does this sugar get into a cell? If the sugar can't go across the membrane of a cell, how does it get in? And it's conventional thinking now is that insulin triggers the production of channel proteins and carrier proteins to allow the insulin to, or the insulin allows the formation of a carrier protein or a channel protein to let glucose come into a cell. Because glucose can't just come up to the membrane and say, hey, I want to come in, because it's not fat soluble. But if insulin has been there first and opened up a tube, a carrier protein or a channel protein, then the sugar can come in. And it's thought that diabetes, we all know there's two types of diabetes, type 1 and type 2. Type 1 is you're not producing enough insulin, or type 2, you're producing insulin, it's not being recognized. In either case, if you don't have enough insulin or you don't have the right insulin, then you're not going to make these protein channels and the sugars can't get into the cell. So, but that's what proteins do for us. They are carriers. They are carriers, they're channels. They allow nutrients and water to get into the cell. They can also be enzymes. These proteins embedded in the membrane can also be enzymes that function as reactive sites. You know what an enzyme does? Remember, you take two reactants and come together and you get a new product. Well, we can have enzymes embedded on the surface of the cell or on the inside of the inside surface. And we also have proteins functioning as plain old receptors for other proteins or hormones. The thing about the protein-based hormones is that they're too big to even try to get in the cell. But if they land on the cell at the right location, they'll activate a receptor that will send their signal down into the cell. So, some proteins are just too massive to even try to get across the membrane. Now, the peripheral proteins, they're the ones that are not going to go all the way through. They're usually on the interior surface, but they can be on the top surface too. Again, they function as enzymes. They function for what we call cell communication. So when a cell has to talk to another cell, we can have proteins that allow this to happen. Our heart muscles have cell connections from openings, literally openings in their cell membranes that go from cell to cell to cell to cell. So we can send an electrical signal across the heart. We can start a contraction by sending an electrical signal from the top of the heart down to the base of the heart and back up again in less than 0.7 of a second, and do it again and again and again. Because the entire one cardiac cycle is 0.7 of a second, and about 3/4 of that time is spent in the rest stage. So we're talking about sending a signal from cell to cell to cell at 300 or 400 miles an hour. Granted, it only has to go about that far because your heart's only about the size of your fist, but it does that over and over and over again. Why do we have holes in the cells? Because it's faster to send that signal through the opening in the side of the cell than it is to come in through the top and go down to the bottom of the cell and back up to the top and down to the bottom of the cell. At 0.7 of a second, you don't have a lot of time to waste, considering you're going to spend 3/4 of that time in a rest phase. Anyway, we also have proteins that function for muscle contraction. Skeletal muscle, the proteins in skeletal muscle slide across each other. So our muscles will shorten when they contract. And then when they relax, they'll go back to their original shape. And we do this all the time. That's what, what we call the motor proteins. They move the cell around. We also have the similar type of, in fact, the same type of protein used in muscle contractions is used in cytokinesis that squeezes the cell to separate into the two daughter cells. So, okay. Now, here's a really, really busy slide that you can read at your convenience. But the proteins, we have at least 6 different categories here. We have transport, signal transduction, cytoskeleton attachment. Cells have their own skeleton too. Enzymes, intercellular joining, cells linked together, and cell-to-cell recognition. So we use these proteins to say, hey, it's me. So, for example, since many of us are more visual than anything else, these are helpful.
Transport proteins carry molecules from place to place. The example shown here allows certain solute molecules to enter the cell. Hemoglobin is the transport protein that carries oxygen in the blood.
Now, most of our transport proteins are going to be involved with moving water into the cell, or water out of a cell, or glucose into the cell, or sodium into the cell, or potassium out of the cell. That's what they do. They don't, the protein changes its shape, so things that can't normally go across the membrane can move through the membrane, and it works very well. Larger proteins like hemoglobin carries oxygen. Hemoglobin's a huge molecule, 360 amino acids long. It's a great big molecule, and it doesn't have to go into the cell. It just has to carry oxygen inside a red blood cell so that oxygen can get released and diffuse into the cell. But it is also considered a transport protein. Now, this second type we see here.
Receptor molecules bind to signal molecules and can then emit second messengers which trigger changes inside a cell. Receptors are thus important links in the system of communication among cells. Some signal molecules, such as hormones, are also proteins.
Now, a signal molecule, a hormone that's a protein, and most of our hormones are proteins. They're big, they're large molecules, and they're not going to be able to go across the cell membrane. So there is a receptor protein on the surface of all of our cells for the different hormones, and it'll land there and activate that receptor protein embedded in the surface of the membrane and say, hey, I'm here, we need you to do this. And they're all specialized. And so the response will be that the receptor protein embedded in the surface of the cell can then communicate to the interior of the cell, and the epinephrine or the growth hormone or thyroid stimulating hormone never has to actually get into the cell. Its message gets in. That's all that matters. We don't. That's why we call these second messengers. Now, since I've said that, you can go ahead and forget about the term second messenger until we get into AP2, because then you'll hear all about it. But because when we talk about hormones, first chapter in AP2 is all about hormones. Okay, now we've got structure.
Structural proteins have many functions. Like tentpoles and ropes, they shape cells and anchor cell parts. They may serve as tracks along which cell parts can move. They bind cells together, making organized units such as muscles, ligaments, and the tendons that bind muscles to bones. The silk of spiders and the hair of mammals are also structural proteins.
Consider collagen. Collagen is the most abundant protein in the body, and it lines up in nice, even rows, side by side by side. It forms the tendons that attach our muscles to our bones, and it's incredibly strong. It's incredibly powerful. It will not break. It'll break bone before it itself tears. So the structure something like collagen is very, very important to us. It helps it gives us that ability to pull on the bone. Our muscles aren't strong enough to do the job we want them to do. But we amplify their strength by using the bone as a lever. So that's how we work more efficiently by doing that. So a lot of our structures, our body structures, are based on these different proteins. And then of course we have, enzymes.
An enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells.
If we're going to break down sugar in our mouth, we're going to use the enzyme amylase. We take take starch, a very long polysaccharide molecule made up of thousands and thousands of glucose molecules, and we'll break it apart, ultimately using amylase, the enzyme amylase, to tear it down into glucose. It's the only place that's where it starts. There are only a few places in our body where we can really start the process of carbohydrate digestion, and it usually starts in our mouth with amylase. Okay, now the glycocalyx. I mentioned this. You'll hear about this again in AP2. The glycocalyx is the identifier. It's a carbohydrate protein combination. Some are attached to fats, but most are proteins. And this is the marker. This is a sugar-coated marker telling us that it's us. These are the biological markers on all of our cells that tell us that you belong, that you belong to me. This is my cell, and don't attack it. So our immune system doesn't attack it because it recognizes the glycocalyx. The markers on our blood cells that determine our blood type, A, B, AB, or O, are also glycocalyx structures. So in addition to recognizing the cell as us, all of our blood cells have another marker on here saying what type blood they are. So so that we don't, and our circulatory system has some very specialized proteins in solution that are looking for the wrong kind of blood to destroy it. So we depend on our glycocalyx structures on our cells to identify us to our immune system. We depend on those structures on our red blood cells so that we donate the right blood or we receive the right blood when we need it. Because giving someone the wrong kind of blood is a really bad thing to happen. A transfusion mismatch, it can have fatal consequences, and sometimes it does. Okay. Something else about the glycocalyx structures that are sort of bizarre. If you have a cancer cells have a glycocalyx structure too. And just about the time your immune system recognizes it as a foreign cell that needs to be destroyed, it changes it. Because our immune system takes about 2 weeks to respond to a foreign antigen, a foreign glycocalyx. So at just about the time that we say, hey, that doesn't belong here, that's foreign, the glycocalyx changes. And then the 2-week clock has to start all over again. So our immune system has a hard time keeping up with cancer cells because they're constantly changing their identifiers. So, and it can reproduce, it can grow. That's what cancer cells do. Remember that cancer is nothing but uncontrolled cell growth. That's all it is, uncontrolled cell growth. And it eventually, if the uncontrolled cell growth continues, it eventually destroys the host. And our immune system can't stop it like it would stop other infections because it doesn't, the glycocalyx keeps changing. We never get a chance to get a handle on it. Okay. Now, cells can either be bound together or they can float free, like red cells or white cells in our bodies, but others are tied together, into tissues. And they are held together 3 different ways. We have what are known as tight junctions, desmosomes, or gap junctions. Tight junctions. The proteins, the integral proteins in 2 adjacent cells fuse together. They're actually, it's like they're stitched with a cytoplasmic sewing machine, if you will. It's like the membranes are stitched together as these proteins extend from one cell membrane to another, and tight junctions are exactly what they sound like. They're very, very tight. We line our stomach with tight junctions so that stomach acid doesn't leak out. It forms a barrier that nothing can penetrate. And so wherever we have a tight junction, we're going to have it's like an impenetrable solid barrier here. It's a very, very strong connection. It's also pretty rigid too. Desmosomes occur, they look more like a zipper because we have proteins that extend from one side to the other, and on either side of this cell membranes, they're sort of locked in place like they have a button attached. Or, the structure in the middle sort of looks like a zipper here. Now, since we use keratin and collagen to make up a desmosome, there's great strength, but a lot of play. There's some flexibility here. We would have desmosomes where we want to have lots of movement, but we want to keep everything held together. The heart cells are held together by desmosomes because the heart has to contract every 7/10 of a second. And when the heart contracts, every muscle cell in the heart goes along with it. Every cell goes or no cell goes. And so it's a very powerful contraction. And the cardiac muscle cells are held together by these desmosomes. Great strength with great flexibility. And the third category are the gap junctions. We see these again in the heart. These are the connections that have holes in there that allow signals, in the case of the heart, to allow the nerve signal that's running through the heart, telling the muscles to contract, can pass through these openings, these gap junctions, deep inside the cell, and not have to deal with going up around the top, going back in. You have to. That's fine. So that's what that's what the gap junctions do. So places like the heart are a great example of where we have gap junctions. Smooth muscle uses gap junctions too, but it's very important in the heart because we have to have this rapid pattern of communication from every cell. We have to start the impulse in the heart up in the upper right atria, up the right chamber, the top right chamber, spread it across to the left top chamber, and then down the middle of the heart, down to the pointy end, the apex, and back up the sides. We have to do that, in under, we have to be done and ready to go again in every 7/10 of a second. And we use these gap junctions here for that to occur. Okay, this is something we've talked about in lab already, so I won't beat this up too much. Passive transport. Cells love passive transport because it doesn't cost them any energy. We have diffusion and we have filtration. We do a lot of filtering in our bodies too. Diffusion can be simple diffusion, movement from a high concentration to a low concentration. Oxygen does that all the time. There's always more oxygen in the blood than there is in a cell. So oxygen always diffuses from the red cell into the tissue. Carbon dioxide always diffuses out. There's always more CO2 in a tissue cell than there is in the blood. So it always goes back out. So that's simple diffusion, always from a high concentration to a low concentration. Carrier mediated diffusion, we have to have a carrier protein or a channel protein. We have to have help getting something into a cell or out of the cell. And of course, there's always osmosis. Movement of water from a high water to low water across a membrane. That's how we're shifting water around inside the cells, between the cells and the interstitial fluid. What about filtration? Well, we filter our blood 60 times a day. Our entire blood supply is filtered through our kidneys 60 times a day. And waste products that we don't want to keep are pushed out through our kidney tubules, ultimately ending up in our urinary bladder, and we pee it away. So, every 45 minutes, we have filtered our blood once, again and again, our entire blood supply. So we do a lot of filtration. Now, the simple diffusion in a cell membrane, the only things that can come across through simple diffusion are going to be the gases, CO2 and oxygen, and anything that's fat soluble. So your cholesterol-based steroid hormones would go across. But simple diffusion only works for just a few things. And I mentioned this before. We talked about diffusion going down its concentration gradient from a high concentration to a low concentration. And we see that diffusion taking place right here in the example on the screen. Put in a dye pellet and the dye is spreading out. Why is the dye moving in here? Because the water molecules are moving. The water molecules are always in motion, just like the air molecules are always in motion. And so they're causing the dye to spread. If we're talking about diffusion in a cell, again, it's air molecule, water, oxygen molecules, or CO2 molecules going from the high side to the low side. We know that smaller molecules diffuse faster than larger molecules. That's why oxygen can go across a cell membrane. And we always go from high to low. It's a free ride, and cells love free. Cells are cheap. I'm sorry, cells are frugal. They're not cheap. They're just frugal. So what do you need to remember about diffusion? Same thing you needed to remember in the lab test, that diffusion only works for small molecules and fat soluble, lipid soluble molecules. If you're very small, you can get across easily. If you're lipid soluble, you can get across easily. If you need to get into a cell, like glucose needs to get into a cell, or sodium needs to get into a cell, you got to use a carrier molecule. So that's an assisted, that's how we have to assist it. So problems with the cell membrane. If the cell membrane is damaged, then we have a real issue with the cytoplasm leaking out of the cells. The cells are made, they're a contained structure. Now, when a patient is burned, severely burned, talking 3rd-degree burn, 3rd-degree burns look like barbecue on speed. We all know what barbecue looks like. You got ribs, you see the rib. Who doesn't like ribs? I hope this doesn't mess up ribs for you. But ribs burned down to the bone is what our tissue looks like in 3rd-degree burns. And that destroys the cell membranes, and the contents of the cells leak out. So your patient with 3rd-degree burns, first of all, they're probably not going to feel most of it because all the pain receptors have been destroyed, but they're going to be oozing fluids. As fast as you're putting in fluids through the IV, it's going to be coming back out. Yes, Emma.
So I actually got 2nd-degree burns from a candle fire on my face,
Mm-hmm.
which you'd never know because there's no scarring. I could hardly feel anything here, and my chin, I could just feel it constantly dripping that fluid that you're talking about.
Mm-hmm.
And I had to have 2 of those IV bags.
Right. Right.
to replace that fluid.
Exactly. You burned away the pain receptors, so it didn't really hurt, but you could definitely sense the dripping, right?
Yeah, I was so much.
Yeah, our patients with that kind of injury lose a lot of fluids. It's hard to maintain their fluid levels. And of course, all that fluid leaking out in a warm, moist environment is a breeding ground for bacteria. So you have some really, it's very critical when we're dealing with burn patients like that. So something as simple as the cell membrane, as boring as the cell membrane sounds, it can cause a real issue in healthcare. So, and no, I don't really get this excited about cell membranes. I'm just trying to keep it from putting you all to sleep. So, because I certainly don't go down to Dr. Grice and say, hey, how about those cell membranes? What do you think? What do you think about cell membranes? Huh? Nobody in our division gets that excited about cell membranes. So, okay, facilitated diffusion. You got to have a carrier protein. You got to have a channel protein. You need help. That's where the term facilitated comes in. You get help to get your sodiums across the membrane. You may have a protein, like the purple thing there, has a specific shape just for sodium. And sodium can go into that channel and slide through and come out the other side. And potassium won't fit there, but sodium will. And you can have one that allows you to let in glucose. And sometimes glucose piggybacks with sodium. So you have all these mechanisms, these carrier proteins and channel proteins that will facilitate the movement across the membrane. Now, water has it easy. Water has a channel protein that's just a tube. We call it an aquaporin. Fancy way of saying it's a water tube. Water goes through. It's a tiny little opening, but small enough for the water molecules to go through, or large enough for the water molecules to go through. But sodium needs more help. And we want sodium to come into the cell. We don't want it to go back out. We want glucose to come into the cell. We don't want it to go back out. Or sugar, we don't want to lose our sugars. And so we have these specialized channel carrier proteins that look like that. And let the substances in. Hang on one second. Okay. So my wife has bronchitis and she's trying to get into the doctor's office. She's just given up and she's going to go to the walk-in clinic over in Middle Creek. So anyway, okay, back to this. The facilitated diffusion then is when you get help to get into the cell. Okay. Now, carrier-mediated facilitated diffusion. This is when you have very specialized molecules. They're going to, like glucose has carrier-mediated. What that means is you have a special protein just for glucose. Nothing else can come in with that protein except glucose. Amino acids may come in this way. These are very special. They can change their shape to, they're closed and a glucose molecule comes up to it and it opens up and the glucose comes into the protein. The top of the protein closes and the bottom opens and the glucose goes into the cell. Or the glucose comes along with its best friend, sodium, and the sodium lands on the carrier protein and lands on the carrier protein, and that causes the carrier protein to change its shape to open up for the glucose. So it's a little more sophisticated than just a channel protein or a regular carrier protein. The carrier protein will change its structure to let something in. And it works as long as we have enough carrier proteins. And that's the key when we get into talking about diabetes, is that if we don't have good insulin or we don't have enough insulin, when I say good insulin, in adult onset type 2 diabetes, the pancreas is still producing lots of insulin, but the cells don't recognize it. The cells refuse to recognize it. The cells have this phenomenon known as downregulation, where they've been exposed to high levels of insulin for a while, and they said, nope, no more. I'm not gonna recognize it. And that's, because, so then we're not getting the carrier proteins we need in type 2 diabetes. In type 1 diabetes, the pancreas just stopped making insulin. In either case, we're not going to get the carrier proteins. We need the specialized carrier proteins to get to open up the membrane to let the sugar come through. Okay. Channel mediated is slightly different. We have leakage channels and we have gated channels. Now, the leakage channels are always open. Leakage channels, water comes in, sodium comes in, potassium comes in, they're always going to open. Sodium is always leaking into our cells, potassium is always leaking out. It's just because of the nature. We've got lots of potassium inside a cell and lots of sodium on the outside. They're going to leak all the time. Now, there's a second type of channel here called a gated channel. These are under greater control. Gated channels are electrical or mechanical. A gated channel will only open up if it receives an electrical signal, like your garage door. You press the button and your garage door goes up. Your neighbor's garage door doesn't go up, yours does, hopefully, and hopefully it goes down too. It used to be, when I was a kid, you could press your garage door opener, and if they were on this, you could set them for the same channel as all your friends' houses, and you could watch all their garage doors come up from one remote. Believe it, yeah. I never got caught. I'll just let it go at that. So, but you could do that because they all had the same channel. One button opened up a dozen garage doors. Now they're all different. Your garage door, if you have a garage door opener, you only open up your garage door and nobody else's, because it's a specialized instruction. Now, we have gated channels in our bodies for sodium and for potassium that are going to release sodium or release potassium, that are going to change the charge on our membranes, and they are either chemically controlled or mechanically controlled or electrically controlled. So some channels, some of these gated channels open up when they get pressure put on them. Some open up when there's a chemical nearby. The chemical is called what we call a neurotransmitter. And some open up when we just change the charge. So when we get into muscle contractions and later on in the nervous system, we'll talk all about these different types of chemically gated, mechanically gated, electrically gated channels. Right now, we're only going to look at the channels that are all passive, channels that leak. Okay, now cells love to get stuff inside or get stuff outside, but we say they are selectively permeable. Some things get in, some things don't. I mean, if you consider, the screen in your window, a window screen is selectively permeable. It keeps out bugs, right? Doesn't keep out dust, doesn't keep out rain, but it keeps the bugs out. So it's selectively permeable, just like a cell membrane. Some things get through, some things don't. Some things pass through very easily and some don't. Some never get across, like the large protein molecules never get across. They have to have what they call those second messengers. Now, cells prefer everything to be free. They prefer it to be passive. Cells love osmosis and diffusion and facilitated diffusion, where we have specialized carrier proteins that just do their job for free. But sometimes a cell has to spend energy to get things into the cell or outside the cell. And those, we call that an active process or active transport. So we have passive transport, which is free, active transport, which requires the use of ATP, the molecule we just talked about earlier in the class. So now, osmosis is passive. That's one of the, water movement across the membrane is a passive process, even though it depends on the solute concentrations. We always go from the, water always goes from the high water side to the low water side. And that high water versus low water is based on the concentration of the solutes in the solution across the membrane. So if we have too much water on the outside of a cell, that water will go into the cell. If there's more water inside the cell than the outside, water will leave the cell and go out into the interstitial fluid. Water always goes from the higher water side to the lower water side, but it's a passive process. This doesn't require any energy. Where we get in trouble is if a person becomes dehydrated. Now consider what happens here. When you become dehydrated, the first place you lose water is in your plasma, because everything we eat and drink ends up in our plasma. So, you're drinking, what is that, a Red Bull?
Yeah.
Okay. Or coffee, like I've got my coffee and everybody's got something. Most people have something to drink here. Everything we drink ends up in our plasma. When we become dehydrated, the first place we notice it is going to be in our plasma levels. We're going to be losing water. Well, plasma always goes out, leaks out of the blood vessels. The blood vessels are not a perfectly sealed tube. They've got lots and lots of holes in them because the plasma has to leak out into the spaces around the cells. That's where we call it the interstitial fluid. And when it does that, it's bringing glucose with it, proteins with it, hormones with it, leaking out from the blood. Well, if the water level has dropped in the plasma, the water level drops in the interstitial fluid. You have less water now around your cells. If your water level drops, it means your salt concentration will go up. So the amount of salt that we have in our body stays pretty constant. And how salty we are is based on how much water we have. We lose a lot of water, we have a lot of salt left behind. We don't have a lot of water in the interstitial fluid, we're pretty salty. So normally the water level inside the cell is the same as what's in the interstitial fluid. But if you haven't had a lot of water for a day or two, then you don't have a lot of water around the cells in the interstitial fluid, you're pretty salty. In fact, you're saltier than the content in the cell. So osmosis says, well, we have higher water levels inside the cell than we do in the interstitial fluid, and water moves out into the interstitial spaces, and the cell starts to shrink. Cells don't like that. When cells shrink, they ultimately can die. Brain cells do not like to give up water. So this is why, water levels are so important to us, why we stay hydrated all the time. A person will die after 3 days without water. You can only go for 72 hours without water. You can go for about a month without food, but only about 3 days without water. And the bad thing about it is the third day, you're pretty much in some sort of psychotic state anyway, so you don't know how to find water or drink it, even if you could find it. So, okay, now. I've already mentioned this in lab. There's a term here, osmolarity. I don't want to beat this into the ground, but osmolarity is simply a measure of the total number of solute particles. Yes, we talk about osmolarity and osmotic concentration. If we have keep this in mind, if we have a 10% salt solution, it's 10% salt and 90% water. A 40% salt solution is 40% salt and 60% water. And water always goes from the higher water side to the lower water side until we reach equilibrium. But in our bodies, it's very, we're never going to reach equilibrium in our bodies. Okay. When cells give up water, they're going to shrink. This hydrostatic pressure is going to be what's driving osmosis. That's the pressure of the water inside the cell. When a cell gives up water, there's less pressure, so the cell's going to shrink. So your cells are going to shrivel up. You know, what happens to a plant if you don't water it? It shrivels up, right? And if you let it go long enough, it dies. You know, so okay, so I kill lots of plants. That's fine. You know, that's what they're there for. But cells give up water all the time. Cells actually in plants are in a losing struggle to maintain water. You know, you forget the water for 2 or 3 weeks, they're just going to up and die. So, but they're always giving up water. They're always sending water to the outside of their cells. Same with us. You know, if we, our cells will shrivel if we get too salty in the interstitial fluid. If we don't have enough water in our plasma, then we're going to suffer the consequences. So, okay.
The plasma membrane is permeable to water molecules, and the movement of water into and out of cells is critical to life. Diffusion of water molecules across a selectively permeable membrane is a special kind of passive transport called osmosis. Plant cells are surrounded by rigid cell walls. When plant cells are exposed to hypotonic environments, water rushes into the cell, and the cell swells, but is kept from breaking by the rigid wall layer. The pressure of the cell pushing against the wall makes the cell turgid and is the desired state for most plant tissues. For instance, placing a wilted celery stalk or lettuce leaf in a hypotonic environment of pure water will often revive the leaf by inducing turgor in the plant cells. Animal cells lack rigid cell walls. When they are exposed to hypotonic environments, water rushes into the cell and the cell swells. Eventually, if water is not removed from the cell, the pressure will exceed the tensile strength of the cell and it will burst open or lyse. Many single-celled protists living in freshwater environments have contractile vacuoles that pump water back out of the cell in order to maintain osmotic equilibrium and avoid lysis. Plant cells are surrounded by rigid cell walls. When plant cells are exposed to hypertonic environments, water rushes out of the cell and the cell shrinks away from the rigid wall. These cells are dehydrated and lose most or all physiological functions while in the shriveled state. If cells are returned to isotonic or hypotonic environments, water re-enters the cell and normal functioning may be restored. Animal cells lack rigid cell walls. When they are exposed to hypertonic environments, water rushes out of the cell and the cell shrinks. The resulting cells are dehydrated and lose most or all physiological functions while in the shriveled state. If the cells are returned to isotonic or hypotonic environments, water re-enters the cell and normal functioning may be restored.
So if you were to take a red blood cell and drop that into salt water, let's say, because red blood cells normally function in a 0.9% sodium chloride, but suppose you put a red blood cell in a 10% salt solution, the red blood cell would shrivel up because there's more water inside the red cell, bless you, than there is in the 10% salt solution. You know, inside the red cell, it's 0.9% sodium chloride. If you drop it in a 10%, it's only 90% water compared to 99.1% water. The red cell will shrivel and try to send its water out into the beaker of 10% salt to try and reach equilibrium. You know it can't, but it will shrivel up. If you, on the other hand, take a red blood cell and drop it into a beaker of distilled water, and we've done this on the microscope, we take a red cell take some red cells and put a drop of distilled water on it, and you're looking at the red cells, all of a sudden there's a red smear because all the red cells have exploded because they've sucked up all the water. You know, they rupture, they lysis because of this. And this is what happens when there's too much water. An individual can get into real trouble by drinking too much water. Because remember, where's everything we drink go?
Or the plastic.
In our plasma. So if you decide that you're going to drink 10 bottles of water this afternoon, one after another, because you lost a bet, you know, okay. So you're drinking 10 bottles of water, those big 2-liter bottles of water, one after another, after another, after another. It's all going to end up in your plasma. And when it ends up in your plasma, it's going to end up around your cells. And what are your cells going to do? Well, they're going to say, hey, the water content, there's higher water outside the cells. We better let it in. And they're going to swell up and they may rupture. And that can be fatal. People can die from what's called water intoxication. Again, if some is good, more is not better. So, and there have been there is a very dramatic case in this where a woman died by engaging in some water drinking contest. So, and I'll pick up with that next time. So, okay, let's get us out of here. And I'll see some of you in lab this afternoon. So.